Good morning, everyone, and welcome to Cardiac Science's Grand Rounds for, what is it, March 17th? St. Patrick's Day. There you go, green. Thank you, Catherine. This morning, we have Tammy Ryan. Kind of an Irishish name.
Yeah,
Yeah?
it is. Yeah, it is. You noticed the green as well?
Yeah.
It wasn't intentional.
Tammy is a fellow in our electrophysiology group, and we are interested in recruiting her back to Calgary as a staff member over the next year. Tammy did a PhD in Ottawa, I believe, and her core cardiology at McMaster before moving on to Calgary to do an electrophysiology fellowship, which she's doing now. Along with her clinical fellowship, she continues to work in the wet lab, and today she's going to talk about that work on stem cell models in cardiac disease. So Tammy, welcome, and thank you for agreeing to present at Grand Round.
Okay, thank you so much for inviting me and giving me the opportunity to speak today. As Dr. Howarth said, I'm going to talk about stem cell models of cardiac disease, but the purpose of today is not going to be to kind of delve in depth into my own work. It's more going to be to give an overview of how we use these models for cardiac development and disease, and I will use some of my own work as examples to put it in context. But it's more of a broad overview of stem cells in cardiac disease. My only disclosure is that I have research funding from Pfizer, which pays for the work that we do on transthyretin, which I'll talk about a little bit today. And the other disclosure is there is a little bit of neuroscience in this talk, but I'll keep it to a minimum, and I promise it is relevant. Okay, so the talk today is really going to be a basic science talk, or foundational science, translational science, whatever you want to call it. And I think sometimes there's a bit of a disconnect between what basic scientists do in the lab and what non-basic scientists think that we do in the lab. And so this was an article that was published in Circulation Research a few years ago. And the title of it was The Growing Importance of Basic Models of Cardiovascular Disease. And from that title, you would infer that that person thinks basic models of cardiovascular disease are important. But in that article, he said the road to failed therapies is littered with interventions that appeared promising in animal models. And I was actually quite offended by this statement, because I think the road to failed therapies is littered with a lot of things, including things that appeared promising in people. And so I think this is just reflective of, perhaps a lack of understanding of kind of how basic science works and how it translates to the clinic. And I think it's important, and hopefully I can illustrate that to you today, for basic scientists and clinicians to kind of work together, because I think we have a lot of success stories, particularly here in Calgary, if you guys are familiar with the CRDS story, where working together kind of leads to really good outcomes for patients, and that's in stem cells and in all of these other models. Some of you may have done some work in basic science labs during your training. Even if you haven't, you're probably familiar with mouse models to some extent, which are probably the most common animal model that we use in cardiac disease development, or really in any other disease phenotype as well. But you may be less familiar with some of the other models that have really been important in gaining understanding of cardiac development and disease. Particularly zebrafish have really been important for our understanding of cardiac development and of regeneration and why we don't regenerate that well in the human heart. And as an EP fellow, I'd be remiss if I didn't mention frog eggs. So, not something you've probably thought of as being important, but they're actually commonly used to model ion channels, the reason being that they're absolutely enormous, which makes them really good for difficult techniques like patch clamping. And then we've always had access to lots of immortalized cell lines that we can grow in the lab from humans, from other animals as well. But the issue with those is that they're generally pretty highly genetically modified. And by making them immortal, you kind of mess with their underlying substrate. And so they also have limitations as models. We've learned a lot from all of these, but at the end of the day, there's always been an interest in trying to understand this from a human perspective, because there are things obviously about people that are different from these animals. And over the years, stem cells, human embryonic stem cells and induced pluripotent stem cells have kind of come to fill this niche. With respect to what we'll cover today, as I said, it's going to be a broad overview. So we'll talk about stem cells in their kind of original iteration, so two-dimensional cultures of stem cells and how we use those as models. But then we'll talk a bit about how the models have evolved to become more complex to try to reflect human organs and human disease a little bit better. So we'll talk about organ on a chip and chimeras, which are essentially hybrid models between animals and stem cells. If there's anyone in the room or online who's familiar with stem cell research, you might say, oh, well, where are organoids? I'm not going to talk about organoids today in the interest of time, and also because my personal view is that organ on a chip is going to replace those in the field. Okay, so with all that said, this is a human embryonic stem cell colony growing on a layer of mouse embryonic fibroblasts, which is how we used to culture these cells. We don't really do it that way anymore. But these cells were originally derived in the early 2000s, and you can see it was published in Science, so a very big deal in the scientific community. But it also made its way onto the cover of Time magazine that year, so a very big deal outside the scientific community as well. And I can tell you that having been a basic scientist for several years, we don't often make our way onto the cover of Time magazine. And so the fact that James Thomson, who derived these cells at the University of Wisconsin, was featured tells you what a big deal this was. And part of why this was a big deal, there's the scientific side of it and the non-scientific side of it, which is a little bit less pretty. So the scientific side of it was that this was the first time we had access to pluripotent cells. And what I mean by that is cells from humans that we could use to differentiate into any cell type in the body. So what's great about these, you can make cardiomyocytes, you can make skeletal muscle, skin, neurons, basically whatever you want to. You just need to know how to do it. And that was something that we had never had access to before. But it also kind of ignited a ethical and political firestorm. And I don't know how many of you may have followed this when it initially was going on. But a lot of people felt very strongly that we shouldn't be working with these cells, that we shouldn't be using human embryos to take cells for research purposes. And very shortly after they were derived, actually the US government under George W. Bush made a policy that the NIH could not fund any new research in embryonic stem cells in any new lines. And from my own personal experience, I can tell you there was actually a guy in my med school class who never spoke to me again after I told him that I worked in these cells. So people feel like actually quite strongly about this. And I'll get into that a little bit more later on. So I said that the good thing about these cells is you can use them to make any tissue type in the body, but that you need to know how to do that. And how we know how to do that is because of the lessons we've learned in developmental biology. So from mice particularly, less so in humans, obviously, and actually a lot from chickens. So when you want to make a cardiomyocyte from a pluripotent stem cell, you start with that cell, you kind of force it to become mesoderm, which is one of the 3 germ layers in the embryo, and then you continue to treat it with the same factors that it would see in the embryo to make a cardiac progenitor and then a fully differentiated cardiomyocyte. One of the limitations of these cells, full disclosure, is that even in their fully differentiated phenotype, with the way we do it now, they are a little bit immature relative to what you would see in an adult human. So they beat in the dish, for example, which is always very impressive to people, but is actually reflective of immaturity and retained automaticity, which is a bit of an issue, but something people are working on. As I said, we kind of do this based on, recapitulating what we see in the embryo. And a lot of the original work that was actually done to derive these protocols for cardiomyocyte differentiation in human stem cells was done in Toronto, but in the lab of Dr. Gordon Keller. And he remains actually amongst the most prolific cardiac stem cell biologists in the world. And he did that based on knowledge and approaches that he had gained doing the same thing, trying to drive hematopoiesis in stem cells. And he obviously is a very good developmental biologist, so knows lots about it. The issue with the original protocols that he derived, which were published kind of in the mid-2000-ish, is that they were fairly complicated and they relied on very expensive growth factors. So you had to buy every protein this cell would see in the embryo, you had to then buy from a company or make it yourself. And that's just not accessible for a lot of labs on a limited budget. And so people have had a vested interest in trying to make these protocols less complicated so that we can use these cells in a little bit more accessible way. So these days, if you want to make a cardiomyocyte from a human stem cell, it's actually pretty easy. So you start with your pluripotent cell in culture, and really you just have to manipulate one signaling pathway, which is the Wnt signaling pathway, which probably a lot of you have heard of and may be somewhat familiar with. So early on, you turn Wnt on, and we can now use small molecules to do this instead of recombinant protein, which is a lot cheaper and a lot more reproducible. That makes cardiac mesoderm. You then abruptly inhibit Wnt signaling, and that can drive cardiac progenitors and immature cardiomyocytes. And then you turn Wnt back on, and that can help your cells mature. So when I started working in this field, I was kind of interested in why this transition allowed the cells to differentiate so efficiently. So I worked on this more from a differentiation perspective, not so much a disease modeling perspective. So this was work that I did in my PhD, and you can see that if you take embryonic stem cells and you treat them with Wnt, you actually get a lot less cardiomyocytes if you put it on at a certain time during differentiation, consistent with what I just showed you. And we were able to show that the reason for that is that Wnt causes a really important transcription factor that's important for cardiac gene transcription to get kicked out of the nucleus. So when it gets kicked out, it obviously can't turn those genes on. And so if you inhibit that process by knocking down this protein here through RNA interference, you get a lot more cardiomyocytes just at baseline, and you inhibit the ability of Wnt to inhibit cardiomyogenesis. So basically, you inhibit the inhibitor. So without getting into this in any detail, because I don't think it's relevant to the talk, this is the model that we were able to describe in that paper. So this just gives you a sense. You can actually interrogate fairly complex biological mechanisms in these cells, and this was the first time any of this had been described. So, you know, these cells are pretty awesome in terms of what you can do from them and what you can learn from them. But as I alluded to, there are some limitations, some scientific, some non-scientific. When these cells were originally derived, obviously we take them from human embryos, and so the genetic background or substrate, whatever you want to call it, is whatever it was in that embryo. So if you want to model a genetic disease, you then have to edit the genome of that cell in order to reflect that genetic variant. And these cells were derived before CRISPR was discovered. So for those of you who are not familiar with CRISPR, it's a technology that we use for gene editing now that's actually quite straightforward. But this was before that, and the techniques that were being used around this time were a lot more challenging to use. And so that wasn't actually that easy to do. And then also the ethical concerns. So, you know, I told you about these issues with funding from the NIH, so that really limited what people were able to do. And you might say, well, you know, you had some cell lines, just work in those. But the issue is that if you only have, you know, 5 or 10 lines, that's your gene pool that you're working within. And so the genetic variability was really quite limited. So interestingly, basic scientists are good at finding ways of getting around things. And we kind of did this in 2 ways. First was that, interestingly, the state of California said, you know, we're just not kind of on board with this. So we are interested in this research. We think it's important. We want to fund it. And so that led to the foundation of the California Institute for Regenerative Medicine, or CIRM. And CIRM is a funding body limited to the state of California that funds regenerative medicine projects, particularly stem cell projects. And it was so successful as an initiative that actually scientists from all over the US left and moved to California to take advantage of this. And as a result of that, people came from all over the world to train in California. It's actually where I did my postdoc. And then more people came from all over the world to stay and work there. And California has, as a result, basically become a mecca for stem cell research because of that. They've given out about $25 billion in funding. They've trained thousands of stem cell biologists and created lots of jobs. And actually, they're still kind of going strong. But obviously, that only solved the problem in the state of California. And so also people started thinking about what are some other ways that we could potentially get access to a pluripotent cell. And that led Shinya Yamanaka, who was a scientist in Japan, to basically ask, could you make a stem cell from scratch? And turns out you can. So he took skin cells from people and overexpressed the same genes that are important for pluripotency in embryonic stem cells and basically turned it into a stem cell. The efficiency of it is kind of low, but we've refined this technique over the years, and it's actually quite good. Again, a big deal. This was awarded the Nobel Prize in Physiology or Medicine in 2012. And he shared that prize with John Gurdon, who's actually not a stem cell biologist. He is a scientist at Cambridge who did a lot of the early nuclear transfer experiments that were required to essentially to be able to do this. So how do these cells work? So you take a person, you take cells from that person, whether it's blood, skin, urine, epithelial cells, really almost any cell type in the body you can do this with. And you just express these 4 genes, and that turns them into stem cells. You then can use them the same way that you would use an embryonic stem cell and differentiate it into any cell type in the body. And the benefit of this is that if you take these cells from a person who has a genetic disease, your cells will also have that same genetic variant. And now that we have CRISPR and we can edit things really easily, you can actually correct that mutation and make what we call an isogenic corrected control, so that your control cell line is the exact same genetic background as your cell line of interest, with the exception that you've corrected that gene mutation. And so people are using these for disease modeling, for drug screening, and also for cell therapy, which I won't talk about at length, but I'll just touch on briefly so that you don't think I'm just espousing witchcraft. So interestingly, the very first IPS, the very first disease to be modeled in IPS cells was actually long QT syndrome. So this group took skin cells from a patient with long QT1 and made IPS cells and then differentiated them to cardiomyocytes and did a bunch of analyses on them. And you can see that in these cardiomyocytes derived from control cells, KCNQ1 localizes pretty extensively to the plasma membrane, which is where you would expect an ion channel to go. But in the patients with the KCNQ1 variant and long QT syndrome, the protein actually doesn't go to the plasma membrane. It stays stuck kind of around the nucleus. And if you co-label for PDI, which is a marker of the endoplasmic reticulum, basically the protein is just stuck in the ER. So it has a trafficking defect, which is why it can't get to the plasma membrane, and that's why this person has long QT and repolarization issues. Since then, there's been probably at this point at least hundreds, if not thousands, of IPS cell models that have been made of cardiac disease. So basically every cardiomyopathy you can think of, all of the inherited arrhythmias have been modeled in IPS cells. And I'm sure you can tell from this talk that I really love stem cells, but I have to say I don't think the existing models of ischemia are super convincing. I think that will change with the advent of organ on a chip and possibly with chimeras, but for now I don't actually think that they're as good as some of our other models. With respect to using these in patients, so these cells are being used in many clinical trials. This is a table that summarizes the clinical trials that are purely cardiac related. So the top half of the table in blue, these are trials that involve just taking cells from patients for disease modeling purposes. But the bottom half of the table in orange, these are clinical trials where these cells are being implanted into people, primarily for heart failure, so to try and restore LV function. I think it's going to be challenging to do this, and if you think about this purely from an EP perspective, so there's lots of EP people here, if you take cardiomyocytes and you just plunk them down into existing muscle, you can imagine that this creates a substrate for arrhythmias, and actually one of the major issues is that if you do this in monkeys, they all get VT, like every single monkey gets VT. So I think it'll be challenging, but I think we will get there. And actually the cardiac field is fairly behind on this. This is quite far along in type 1 diabetes and in Parkinson's disease, where these cells are being actively implanted into people and are actually showing benefit. No, so the the diabetes ones, they make like a little kind of pancreas, a little fake pancreas, like in a cartridge, and they put it inside the person. Or sometimes they inject it directly into the pancreas. There's lots of different ways you can do it. For Parkinson's, they actually do like stereotactic injections of the cells into the relevant region of the brain. So it's for this extremely challenging. For cardiac, it's only really been, I don't know all of the protocols for the different clinical trials that I showed. Mostly it's not systemic. So sometimes what we do, not we, but they, is take like a biomaterial or a hydrogel and actually incorporate the cells into that and use that as a substrate to attach it to the heart. But again, you have this kind of automatous functioning area of cells that doesn't always get on board with what's going on around it. So when I started working with iPS cells, I actually started doing it in Parkinson's disease. So this is where we take a little bit of a neuroscience turn, but I'll keep it short. And we have cells in the lab that we use that were taken from patients that have genetic variants in alpha-synuclein, which if you remember from med school is a component of Lewy bodies in PD. And we differentiate those cells to dopaminergic neurons. We also have an isogenic corrected control. And we look at what goes on in the cells to try to understand Parkinson's a little bit better. And this image just shows cells that have been differentiated to fully terminally differentiated dopaminergic neurons. If you look at these cells, so we have two mutant cell lines, so E46K and A53T. They actually exhibit a lot of the same pathological features that you would see in a Lewy body or in a patient. And that's shown here by phosphoserine 129, which is a modification of alpha-synuclein that occurs in Lewy bodies. So we see that in the mutant cell lines, both in ES cell-derived ones and in iPS cell-derived ones, but not in the corrected controls. And we were able to show in this paper that that results in a lot of mitochondrial dysfunction, mitochondrial fragmentation. And the problem with that is that lipids in the mitochondrial membrane are actually required to keep alpha-synuclein from misfolding. And so when you get mitochondrial dysfunction, it actually then perpetuates the problem. So the reason that I think this was important to lead in is this is kind of how I started thinking about could we model cardiac proteotoxicity in these cells? And by that, I mean cardiac amyloidosis. So the way biochemists think about amyloid and the way clinicians think about amyloid is very different. So I think a lot of clinicians think of amyloid as you get a fibril, it deposits in a tissue, that tissue gets messed up, and that's amyloidosis. But in fact, it's a lot more complicated than that. And transthyretin amyloidosis is very similar to Alzheimer's, very similar to PD in the biochemistry of how it works. So as a basic scientist, when you're thinking about amyloid, really we think about it as misfolded protein that has a beta sheet that wasn't supposed to be there. And so we think about the pathophysiology of this differently. With respect to transthyretin, I think probably most of the clinical people in the room know this, but for anyone who's not familiar, it's a protein that's synthesized in the liver, it's then secreted into the circulation, and its job is to carry or transport thyroid hormone and retinol binding protein. For reasons that we don't really understand in the context of aging or in genetic variants in TTR, that tetramer that it forms and when it's happy becomes unstable and falls apart into monomers. Those monomers then become misfolded and stick together into what we call oligomers, which are basically just blobs of misfolded protein. And then those organize to form fibrils, which deposit into the heart and kidney, peripheral nervous system, what have you. And so this is part of how we think about this a little bit differently. So the literature from Parkinson's and Alzheimer's, Huntington, a lot of these misfolded protein disorders actually suggests that what's toxic is the oligomer because it's still floating around damaging things as opposed to the fibril, which is kind of, sitting in the tissue, not fully inert, but is certainly not, perhaps as damaging necessarily as the oligomer. It probably does act as a surface on which new oligomers can nucleate, so it's not benign, but it's probably not the only toxic issue in these diseases, which is probably also part of why some of these mAbs that are being used for Alzheimer's, this kind of push to clear protein aggregates in these diseases, probably a big part of the reason why they don't really work that well. Another important thing to take away from this is that these work like prions for all intents and purposes, if that's how you want to think about them. So this misfolded transthyretin monomer will go around and basically template its misfold onto surrounding transthyretin monomers. And so it basically perpetuates the problem, and that's how you end up with more and more misfolded protein. And that will be relevant a bit a couple slides from now. So in terms of modeling this in stem cells, this is a bit more challenging than Parkinson's disease. So Parkinson's disease in dopaminergic neurons, alpha-synuclein is made locally. It stays inside the cell until it doesn't, and it actually can spread disease from cell to cell, but the initial pathology is intracellular. For transthyretin, as I said, it's secreted into the blood, it's floating around, it comes from a different organ, and it actually deposits into the extracellular matrix outside the cell. And so it doesn't really make sense to just take transthyretin and put it onto cardiomyocytes because you're not actually looking at what's going on in disease. And the interaction with the extracellular matrix is actually quite important. So TTR is known to interact directly with perlecan, which is a component of the cardiac ECM. And patients who have amyloidosis actually have higher levels of expression of some of the factors that are important in the cardiac extracellular matrix. And the ECM in the heart is actually made by cardiac fibroblasts, not by cardiomyocytes. And so that suggests that the toxicity of TTR is exerted on both cardiomyocytes and probably cardiac fibroblasts as well. So that led us to want to design a model that kind of mimicked what we see in patients. So we did this by combining cardiomyocytes and cardiac fibroblasts. So these are cells that I differentiated in the lab. You can see that they go from iPSCs at day 0 to progenitors at day 9, where they express NKX, and then to fully beating cardiomyocytes in the dish by day 17 and remain beating for several days after this. We then combine those with cardiac fibroblasts that we also differentiate from iPSCs, and that's shown here by expression of vimentin. The idea being that those cardiac fibroblasts would secrete extracellular matrix, which would then surround the cardiomyocytes and give us sort of a substrate similar to what is seen in the human heart for these experiments. And then our experimental paradigm is that we use this co-culture to treat the cells with the different forms of transthyretin, whether that's wild-type tetramer, a monomer, which we generate by using 2 genetic variants that prevent the monomer from forming a tetramer, oligomers or fibrils, which we can actually make in the lab. So I'm not going to get in depth into this, but just some of the preliminary work that we've done on this is pretty interesting, I think. So if you take these co-cultures and treat them with transthyretin, and then look at whether or not it sticks to that extracellular matrix by labeling for perlecan. If you treat with wild-type transthyretin, which is essentially always a tetramer, and then label for perlecan and TTR, the transthyretin really doesn't stick. It just gets washed off, when you remove the media. As opposed to if you treat with the monomeric variant, you actually get pretty extensive deposition of the protein in close proximity to perlecan. So we think this is potentially reflective of early events of how this protein comes to deposit and exert its toxicity. As I said, we make these fibrils and oligomers in the lab, and the way that we've done this in the past with alpha-synuclein and now with TTR is you basically just put a whole bunch of protein in a tube and you shake it. And you didn't used to be able to do this with TTR. You had to lower the pH to 4 and do all these ridiculous things that led you to believe that what you were working with was God knows what blob of protein. But now there are new protocols for doing this that are actually pretty good. So these are electron micrographs of fibrils that we've made in the lab of TTR and of oligomers. But if you do this the way I described, which is just take a bunch of recombinant protein that you purified in the lab, put it in a tube and shake it, you can make a fibril and you can make an oligomer, but you really don't know if what you've made is in any way reflective of what is seen in the patient. And so that comes back to what I was saying before about the idea that the misfold is templated from that original protein onto the proteins around it, like a prion. And so what we're working on now is trying to make use of that knowledge to try to generate higher order transthyretin structures that mimic what's seen in patients. So I have to say here a very big thank you to Noel Fine, who has been really instrumental in helping us recruit patients from whom we've managed to purify transthyretin. This is a Western blot for TTR. And you can see here that we have several different species on this Western blot. So this smaller species is likely a monomer. This kind of middle area is likely tetramers and dimers. And then we do see quite a lot of higher order species that we think are reflective of circulating oligomers. And so our next step is to use these, combine them with recombinant protein, and basically try to make oligomers and fibrils that are like what we see in patients. So I'm going to switch gears a little bit now and talk about some more complex models, and that's specifically organ on a chip, which some of you may have heard of. It's kind of a new kind of hot area in stem cell biology. It's actually not that new in the sense that it's been around for 10 or so years. And it started off being fairly crude, whereby people were 3D printing these chips and putting biomaterials on them, and then putting cardiomyocytes on them and trying to see if they made some more organized structures compared to what we just make in the dish. And this is an early example of that, and you can see that on this biomaterial, the cells do beat in an organized fashion. But this has very quickly become significantly more sophisticated. Again, actually, a lot of this work was done in Toronto in the lab of Dr. Milica Radicic, who developed this thing called BioWire. You can't really see it that well because the beating here is not quite as vigorous as some of the other videos, but they are beating, I promise. So she cultures these cells on these special substrates and can actually paste them, can expose them to different toxins, but also different external pressures, and actually has shown that using different techniques, you can mature the cells a little bit more. You can model things like hypertension, like you can do a lot of cool stuff with this. And then she has continued to kind of develop this into these cardiac tissues. So this is the second iteration of it called BioWire 2. And so these are just little cardiac tissues that beat on these wires, and again, you can do some neat stuff with it. But this has now kind of evolved even more into where people are pursuing this from a microfluidic perspective. So this chip on the right is a chip that people are using fairly extensively in the lab now. And the idea is that you have these two channels in which you can have two different types of cells, for example. You can then hook up this chip to a microfluidic pump, very similar to the pump that the anesthesiologists use in the OR for all intents and purposes. And you can flow the media over it. You can put toxins in it. You can control the pressure. You can basically do whatever you want. And so people are using these to make fairly extensive organ models. It's very advanced in the intestine field, I can tell you that. We have done a little bit of this in the lab. This is some work that was done by Kadar, a former grad student in the lab. So he 3D printed one of these microfluidic chips, and he put in one chamber wild-type dopaminergic neurons, and in the other chamber Parkinson's disease dopaminergic neurons, and looked at what happened between the two. So the cells are in two different chambers, but the media can cross between the two. The wild-type neurons are labeled in green. The Parkinson's disease neurons are not shown here. But what you can see is that when you put these cells together, they're not actually touching, but the media is going back and forth between the two. You actually start to see aggregated alpha-synuclein in the wild-type cells. So this gets back to what I was saying about you can actually perpetuate disease from a diseased cell to a healthy cell. And so this is one of the ways in which we can use this chip. But I have some ideas of how to kind of do this with transthyretin that I think will be quite interesting. So one of the reasons that people think that the tetramer destabilizes initially in amyloidosis is that it's differences in shear stress. So that tetramer is exposed to a different level of shear stress, and for whatever reason, that results in it getting destabilized and falling apart. And I think this is actually a great model in which to look at this. So the idea would be to use this microfluidic chip, put cardiomyocytes, cardiac fibroblasts in it, flow media over it. By varying the rate of flow, you vary the rate of shear stress, and then you can look at destabilization of transthyretin and deposition and how that destabilization is affected by changes in shear stress. The last thing I'm going to talk about is chimeras. And interestingly, we have kind of come full circle to the initial story I told you, whereby when stem cells were initially derived, people were kind of, really concerned about the ethics around this, and it led to a lot of restrictions. And I will talk about chimeras, but just to preface it, there actually is a moratorium on doing this in the United States right now. In Canada, nobody seems to have paid as much attention to it. But we'll get into a little bit about the ethics of this. So how to kind of think about this. So we've used mouse models forever and have gained so much valuable information from them. But there are some differences between mice and humans with respect to the action potential and how the heart beats. Obviously, mice hearts beat a lot faster than human hearts. And they use, particularly for outward currents, a lot of different channels. And that's part of why their action potential looks quite a bit different. And so if you think about that in terms of modeling things like inherited arrhythmia syndromes, that can pose challenges. So for example, in mice, the major repolarization current is ITO, and to a lesser extent, IKS. Whereas in humans, we know it's IKS and IKR. And so if you want to give a mouse long QT, you can introduce variants into ITO, but it becomes then a little bit more challenging to relate that to humans. And so people have used iPSCs since they've come out fairly extensively to model inherited arrhythmias. This is from a paper that I wrote with Jason Roberts a few years ago, just outlining all the mechanisms of inherited arrhythmia that have been illustrated in iPSCs. And you can see that there's just a huge variety of stuff here, like trafficking defects, differences in mRNA stability, proteasome and ubiquitination and degradation changes. And all of this is actually fairly well understood. And actually, I have to say, a lot of this was known before. Like, a lot of this was done in frog oocytes and was just confirmed in iPS cells. And a lot of it also we learned from mouse models. But at least one of these has actually led to a clinical trial. So lumacaftor, which is a small molecule that can actually rescue translocation of these proteins to the membrane in a way that is analogous to how the drug that's used in cystic fibrosis rescues trafficking of CFTR, is actually being trialed now in patients with long QT to see if it helps with restoring normal repolarization. But long QT is a little bit different from Brugada. So long QT, there's the three main types. We know the genes associated with each type. The genetics of Brugada syndrome are a lot more complicated. So about 20 to 30% of patients have a variant in SCN5A, but the rest of the other 70 or 80% probably have a polygenic substrate. It's probably not that there are single gene variants for Brugada that we haven't discovered yet. It's probably that it's just a mix of mutations in different genes. And that's reflected here in a GWAS result from patients with Brugada syndrome. So people, though, have still used iPSCs to model Brugada, and that's shown here from the same paper, same type of figure. And you might look at this and say, well, there's lots of stuff on here, just like the long QT image that you showed us. But actually, the data here is not nearly as robust as what people have shown for long QT. Certainly, there's very good evidence that Wnt is important in Brugada, but Wnt is basically important for everything. So that's very low-hanging fruit. And part of the issue is that people, for whatever reason in this field, have chosen to focus on these ultra-rare variants, and actually a lot of times on variants of uncertain significance, or even on genes that have been essentially almost disproven for all intents and purposes as being causative in Brugada. I don't really know why this is, but there are lots of known variants in SCN5A that have never been modeled, but this is an exhaustive list, at least at the time of publication, of all the iPS cell models of Brugada, and you can see that actually the majority are VUSs, so this is an issue, obviously. So then you want to think about how these other 70 to 80% of people who have a polygenic substrate, you can still make an iPS cell from that. You don't actually need to know what your genetic background is. You just still make the cell, and you can still do the experiment. You just don't know what the genetic variants are. And somebody actually did that a few years ago. They took 4 different Brugada patients, made iPS cells from them, differentiated them to cardiomyocytes, did a whole bunch of EP testing on them, and the cells were totally normal. There was nothing wrong with them. And interestingly, I think this fits into what we're learning about Brugada, which is that whereas we initially thought it was a pure channelopathy, we're now learning from MRI data that there's likely a significant structural component. And previously you either subscribed to the depolarization hypothesis or the repolarization hypothesis of how Brugada worked. I find these quite difficult to understand, so I'm not going to try to explain them. Also not relevant to today's talk. But given that the structural substrate seems to be concentrated in the right ventricular outflow tract, that's led people to ask whether this might be related to a population of cells called the neural crest. The neural crest is an area in the embryo adjacent to the neural tube. And what's cool about these cells is that during development, they actually will leave the neural crest and they go all over the embryo and they contribute to tissues all over the embryo and totally unrelated tissue types. And one of the structures that they contribute to significantly is the RBOT. So then you're left with this question of, in Brugada, you have a polygenic substrate, which is not ideally modeled in mice because it's hard to make a mouse model if you don't know what your genetic issue is. And then you have a structural substrate, which, in spite of evolving complexity of these models in iPS cells, is not great in stem cells. And so then that led to the question of what if we combine the two and try to use them together? And this is where the idea of chimeras comes from. So chimeras really are a figure from Greek mythology originally, which was a lion with a goat's head growing out of its back and a snake head for a tail. But developmental biologists have been using these for like 40 years. And in developmental biology, it's any organism that has tissues from genetically distinct individuals from grafting tissue between embryos. And how people have used that is these are quail-chick chimeras. So this is a chick, and this is a quail, and this is a chimera of a chick and a quail. And people basically use these to fate map cells to look at where they go in the embryo. And this is like literally dating back to like the 1980s. This has been around forever. But with the advent of human iPS cells, people started thinking, could we go back to this? And people were thinking about it mostly from the perspective of organs for transplant. And so the idea behind this would be you would take human iPSCs, you would inject them into embryos of, for organ transplant purposes, probably a pig. And then the offspring would have organs from a human inside the pig, which could then be harvested for transplant purposes. Obviously, you can see how this could create some ethical issues. If you inject human stem cells and end up with a pig or a mouse with a human brain, that like freaks people right out, which is understandable. So this is why the moratorium has been placed in the United States. And so the NIH does not fund currently any blastocyst complementation work. But it has not stopped people from exploring the concept, just not using human cells. So in this paper, this group injected rat stem cells into mice and tried to show that the rat stem cells could populate the entire cardiovascular system in the mice, but turns out they can't. So it goes sort of part of the way, but not the whole way, and the embryos don't, make it to fruition. And so this kind of is suggestive, right? Like if a rat stem cell can't even replace a mouse stem cell, how is a human stem cell going to provide the cardiac output that a mouse requires? So, this is probably, at least with our existing technology, not something that you could replace the entire mouse heart with human stem cells, but what you can do is replace the neural crest. These are two papers from the lab of Rudolf Janisch, who's probably one of the best stem cell researchers in the world. And he differentiated iPSCs to neural crest and showed that you could inject them into mice, and they would actually go to these different sites of importance and contribute to those tissues. And so this is something I'd like to explore in my lab once it finally gets up and running. So the idea would be that you would take cells from Brugada patients, both polygenic and SCN5A positive variants, differentiate them to neural crest cells, inject them into embryos, and allow them to then populate these tissues. So the idea then would be that you would end up with a mouse heart that is populated with human cardiomyocytes, specifically in the RVOT. And so this would allow you to model the migration of these cells, how they contribute to the RVOT, track how they potentially contribute to the fibrosis that we see in these patients on MRI, and also look at hopefully the basis for arrhythmia in this system. So that's kind of everything I wanted to cover today. In summary, I hope I've convinced you that HIPSCs and ESCs to a lesser extent offer a good model for looking at human disease, particularly proteotoxic conditions and polygenic disorders where mice and traditional animal models are maybe a bit more challenging. With respect to clinical correlation recommended, I think this just comes back to what I was saying about Brugada. I think it's important as basic scientists that we think about, how we can contribute to patient care. And so I think, trying to focus on potentially areas where we have more evidence that what we're doing leads to differences in how patients are treated, I think can be helpful. And hopefully in the near future, these hybrid models can potentially help us where our traditional models have not been super helpful. And then I'll just end saying thank you to the lab at the HBI, so Dr. Scott Ryan, Shahani, who I have to really say, has saved my lab life. So I'm on green this week, and she was emailing me spreadsheets yesterday at 10 p.m. Because we had a time point yesterday that I couldn't do, so she's just the best. Gerardo, who's doing a lot of the transthyretin work, Noel Fine, who's been super helpful with helping us recruit patients, and then at McMaster, Katie Connolly and Jason Roberts, who have been extremely helpful. And then in the US at UT Southwestern and at the Amyloid Center in Boston, these groups have been very generous with sharing their autopsy-derived transthyretin seeds. So we're very grateful for that. And I'll end there. Oh yeah, chat. I think someone just said they
chat and unmute here. Thank you very much. We will open it up to questions, both in the room and online. If you're online, you can put them into the chat. We've got the window open, or just speak up. So we'll open it up. Catherine, of course.
Fantastic talk, Tammy. Really enjoyed that. And it opens it up to so many different questions, okay? I have several, but I'll start off with one. The work you're doing with Noel Fine and taking the cells, these are from patients that have the disease, right?
No, so we don't actually take the iPS cells from TTR patients. The reason we probably will end up doing that, but for a reason that I didn't get into. We don't mind so much just working with wild type iPS cells because the transthyretin is coming from away. So you don't need the transthyretin to be locally produced. And in fact, cardiomyocytes don't make TTR and neither do cardiac fibroblasts. So for now, we're using recombinant protein. But one thing that we've talked about with the organ on a chip, and I didn't get into this, would be actually putting liver cells into the chip. So then the liver cells make the TTR, which is then secreted. And because we're also looking at this from a neuro perspective in the brain, TTR is made by the choroid plexus. And so actually trying to make choroid plexus cells, put those into the chip and have it circulate in a model of basically CSF for all intents and purposes. And we think that will be more representative of obviously what happens in real life. The issue becomes that if we do that, we don't have a good way of triggering oligomerization or destabilization, except if we can show that the shear stress is an issue, or if we find a mechanism, it's a good way to test it. But for now, because we don't sort of have that setup working, the protein comes from TTR patients, but the cells are just wild type cells.
Okay. So I was wondering if this eventually could become a screening test, just like we test for cholesterol and things like that, but down the road, if this would become a screening test so that we could treat patients early if we see an abnormality.
Yeah, so actually, and this is not to do so much with stem cells, but it's something that we're looking at in PD, and it's from a purely sort of more protein biochemistry, biophysics perspective, looking at what are the early structural changes and whether those can be detected in the CSF. And what I showed you on that Western blot, the fact that we can detect those oligomers floating around in patients, I actually think that is where the diagnostic possibility lies. So these people obviously are being seen by Nulfine. They're about to be started on tafamidis. They have full-blown amyloidosis. But what I'm kind of hoping is that those oligomers are potentially detectable in the blood earlier. And we know that, TTR destabilization, independent of whether you have amyloid, if you have lower blood levels of TTR, you're at risk of dying early. Like, it's actually not an insignificant issue. But what those tests don't show is what the structure of the protein is. They just show the levels. And so I think knowing more about the structure, and you can do this without an iPS cell even, is potentially of diagnostic value, and it's something we're thinking about and working on.
Rob.
Yeah, nice to talk, Tammy. Thanks very much. So my question's about the work where you're using the iPSCs to make both myocytes and fibroblasts. So, of course, I'm sure you're aware that, differentiating iPSCs into fibroblasts, the conditions that you have those cells in will have a big impact on whether they're kind of quiescent or more like activated myofibers and whatnot. So
Yeah.
the question is, how are you handling that, and what have you seen in terms of deposition of matrix, and does it look like what I call a normal amount of matrix, or what is the phenotype of the matrix that you see in these experiments?
Yeah, we haven't looked extensively at the matrix yet, and we haven't tried to activate them to make myofibroblasts yet. That's something, again, I'm hoping to do with the chip. So for now, we're just using like baseline normal cardiac fibroblasts secreting a normal matrix. And we haven't looked at it extensively to see, how it might compare to the matrix that you see in the heart, although I think that's a great idea. But I have thought about this, the idea that, for those of you who are less familiar maybe with cardiac fibroblast biology, so cardiac fibroblasts, are a huge component of the heart. They secrete the extracellular matrix, but they also become problematic in that they get activated to form myofibroblasts, and they secrete too much matrix, and that's how you end up with fibrosis in the heart in various conditions, AFib, after ischemia. And so with respect to Rob's question, he's basically asking, have you thought about this second sort of component of the fibroblast? We have thought about it, but we haven't looked at it yet, but it is on our agenda, because I do think if the TTR is exerting toxicity, I'm sure there's some degree of activation and conversion to myofibroblasts, but we haven't looked at it yet.
Tammy, great talk, really exciting stuff. I'm looking forward to seeing where you're. And with the labs on a chip, how much control do you have over the sort of physical structure of the tissue? Because it's a fairly homogeneous plate of cells across the whole chip, or can you define areas where you could have more fibrosis and cardiomyocytes? Because as you know, in arrhythmia production, and maybe it's relevant for Brugada studies as well. Can you tell part of the chip to be more fibroblast than myocytes, and look at that?
Yeah, so the companies who make these chips, so you can make your own chips, or there are companies that make them. If you make your own, you can define all of that. So if you know, for example, there's a substrate that's more amenable to cardiac fibroblast deposition versus cardiomyocyte, you can control all of that. If you have a company make it, you can ask them to do that for you. So you have basically total control over how the chip is set up. You have control over how many compartments you have, whether those compartments allow, for example, what I showed you with the neurons, whether they allow neurites through or don't, whether they only allow vesicles through, like what the pore size is. You have control over the flow rate over the cells, so that's how you control shear stress. You basically can control every component of it. With respect to what you're getting at for controlling the borders between populations, you could do that with a hard border, meaning you have two different channels, which is how we did the experiment with the dopaminergic neurons. But if you wanted them to be able to touch each other, you would sort of need to set up some sort of, not necessarily a barrier is not what I'm getting at, but potentially two substrates that kind of come together. And you certainly could do that, you just would need to know what it is. So the answer is you could do whatever you want with these. They're pretty you can buy them as plug and play if you don't know what you're doing, or you can make them to be to be incredibly sophisticated.
Along the same line, do you need the fibroblasts, or is there something else in the medium?
So that's actually something that we're interested in from the perspective of what is the role of the extracellular matrix. So I told you that the fibrils kind of deposit in the ECM, and people really want to clear these fibrils, and it seems to be sort of where the clinical world is moving toward. But as I said, we don't actually know that they're super toxic. And in that EM picture that I showed you, by the time you have all those fibrils, those cells are dead. And so trying to clear the fibrils from there, I don't actually know how valuable it is. And so part of what we're thinking about when we're designing these experiments is whether the fibroblasts and the ECM are protective or whether they potentiate toxicity. And what I mean by that is that when those monomers deposit in that ECM, is that actually protective in the sense that it's pulling the monomer away and preventing it from exerting toxicity on things? Or is it toxic because it's pulling it down, it then stays in the tissue and is able to mess things up? So the question I don't think is so much whether you need the fibroblasts, it's what the fibroblasts and the ECM that they secrete are doing in the disease. And that's part of what we're asking, and I don't know the answer yet.
So that was great. 10% that I understood. So phenocopies. Right? The GWAS that you showed briefly of Brugada actually kind of caught my interest, right? Because of the p-values in Brugada, they're kind of arbitrary. You hope you hit one out of the park, and that the others, you want controls to look like, Regina plots and not like Edmonton plots, where there's a hint of a building. So how much of phenocopies do you think are actually polygenic risk copies? Where you've actually got 3 or 4 or 5 genes in there. That's number one. And there's probably no answer, and then the second is, if you've got a polygenic phenocopy, where all the genes, and this is a broader question, all the genes are sort of important, what's the chance of finding a silver bullet directed at one of them that would fix the problem? So that's the first question.
Yeah, I think the silver bullet, the chances are like 0. Honestly, I think these polygenic things, we don't even really have silver bullets for a lot of the single gene disorders. And so the idea that we could find a silver bullet in a polygenic condition, I think is not, I wouldn't be very optimistic about it. And phenocopies, polygenic, I think probably a huge proportion of them are. Are we kind of maybe misdiagnosing some of these people as having these conditions when they have something that's very similar but maybe doesn't meet the exact criteria? Probably. But is it sort of more of a spectrum, and that spectrum is generated by the polygenic substrate? I think that's probably the case. I don't know. I don't think the evidence is there to say that for sure, and I don't think anybody has done that, at least that I'm aware of. Like even that GWAS data from Brugada is just really from the last couple of years. So trying to relate that to phenotype, I'm sure people are doing it, but I don't think that's been done, or at least published that I know of.
Second question is actually not cardiovascular. It has to do with the synucleinopathies. And as you know, sort of the phenotype can kind of wander over the years. Do you have a sense of why that is?
From a purely synuclein biochemical
It
perspective?
start out looking like primary autonomic failure, and then, oops, it's actually Parkinson's 10 years later.
Yeah.
It started looking like early Parkinson's, and then, oh my God, it's Lewy body dementia. What's going on there? Do you or anybody you're close to have a sense?
This is probably a better question for Scott, but I'll try. So there is, I think part of it is the cell type that's affected. In pure Parkinson's, that's dopaminergic neurons, substantia nigra. A lot of those other conditions have involvement of cortical neurons and other cell types, so that's part of it. The other thing is Parkinson's, I think, we're kind of starting to understand that Parkinson's is much more systemic than people had previously understood. So I'm sure, as you know, because I know you love synucleinopathies, a lot of the early phenotypes of these people are like GI stuff, stuff that like people miss and never think of PD, because why would you? But actually there is synucleinopathy in those organs as well. So that's the other part of it. I think, in terms of, I think it gets back to kind of your phenocopy question, and not so much from a polygenic substrate, but I think it's differences in how the protein deposits, and as to why it deposits in those different cell types at different rates, potentially in different people, I don't know. Actually, we're kind of interested now in looking at the cardiac phenotype in PD, of which it does seem that there's a significant connection there. So yeah, I think it's a bit different from the inherited arrhythmias, even probably in the genetic synucleinopathies. But I don't know the answer for why it deposits in certain cell types differently. I will say, I kind of showed you some data, and there's a lot of data out there that there's propagation from cell to cell. And it also may be that in different settings, that propagation ability is different depending on what the underlying genetic substrate is, or where the disease starts to deposit early on. But a concrete answer to what you're asking, I don't think it's necessarily known.
Well, thank you very much, Tammy. I think we'll pass the